Hello, I am Saman Aryana. I'm the Occidental Chair in Energy Environmental Technologies, and I'm a member of the Faculty of Chemical Engineering at the University of Wyoming. I will be talking about transport in complex permeable media. And by permeable media, I have the subsurface in mind. Subsurface, we're talking about really large systems where transport happens at a very fine scale. So we need to understand they transport fluids, CO2, water. If it's petroleum systems, we're thinking about natural gas or oil, hydrogen, CO2, whatever the fluid is that's moving in the subsurface. We'd like to understand the physics of their mixing and interacting at a very small scale inside the pores, and how to relate that understanding across scales to large-scale systems that allow us to be predictive and engineer these systems efficiently. Now, why does this matter? As we're dealing with global climate change, one of our tools in our toolkit is sequestering CO2 out of the atmosphere, out of industrial processes, and storing them in the subsurface for very long periods of time. That requires understanding the movement of CO2 in the context of the subsurface. Additionally, in our energy portfolio, we are interested in being able to store intermittently H2 hydrogen in the subsurface, produce it back, and then use it in industrial processes as a source of energy. Or a still big portion of the energy portfolio consists of the petroleum-based systems. So we're still interested in understanding the movement of natural gas and oil in the subsurface and how to efficiently produce them and use them in industrial processes. Regardless of what the fluids are in the subsurface, it boils down to understanding their movement in the subsurface and being able to capture that understanding at different scales and translating it into predictive descriptions. So I am interested in these systems because of that complexity, the movement at a very fine scale. And there are a number of scales present, from nanometers, so that's a billionth of a meter, to micrometers, which is a millionth of a meter, or larger. And eventually, this movement is then happening in large systems that are at a scale of kilometers or miles. And we need to engineer them at that scale so we are effective. And we do this by having two different processes, two different tracks, research-wise in my lab. We focus on collecting experimental data and basing our understanding of the physics on the observations from the lab. And we do this by exploring the development of nano or microfluidic systems, where we can explore data or explore the movement of fluids in the context of this platform and collect data, make observations, and base our understanding of the physics on these observations and produce models that then we solve and compare the solutions of the models to the experimental observations to see if they're predictive. So first, I'll focus on the observations. The movement in confined systems has been of interest for a very long time. An early example of this was experiments that looked at something called helishov, which consists of two plates of glass that are put right next to each other, and the movement of fluids in the little space, small space that's left between the two. Now, that is of interest to look at the interfaces between fluids. However, if you're interested in looking at pore spaces, one early example of creating pore spaces was the use of bead packs of glass in different confined spaces, and then we look at the movement of fluids inside the spaces between the glass beads. And this can be done very easily optically using cameras, where you take pictures, and the delineation between the different optical responses of the different fluids help us to understand what happens. If you'd like to have more complex network of channels, then one way is to then etch this, create a representation of a complex network, and then somehow etch that network onto the surface of a piece of glass, and then isolate the channel by attaching another, let's say, blank, very undisturbed piece of glass on top. Now we have an isolated network of channels between the two, and then we can inject different fluids inside. With the camera on top, we can take images and get a sense of how the fluids move and mix. And so here you see an example, early example. This is several decades old, of a fluidic system where the pore spaces are complex in their geometry, but they're really large. They're millimeter scale. So in my lab, we work on making devices that have pores at the scale of microns, micrometers. And we also look at pushing the design and fabrication at the edges to create more resilient systems, either miniaturized networks or also devices that are capable of withstanding higher pressure and temperature conditions. An early example of this was our efforts to create devices using sapphire because of its superior mechanical properties. We used femtosecond laser pulses to etch channels on the surface of sapphire, and then we looked at the use of the pulse to also bind a sapphire glass to the one that bears the network. And so we did some experiments with that and got a sense of how to do it. It has its own issues. Then we looked at miniaturization of the network by, instead of doing chemical etching, where we take a piece of glass and through a process that I'll describe briefly here in a few minutes, we then etch or eat away the surface of the glass at very specified locations, pre-specified locations, to create a network of channels. We looked at the use of ion beams, where we bombard the surface of this glass with ions, or we could do it with electrons to go down to nanoscale channels on the surface, and we'd have to find a way to isolate them and then collect data by moving fluids through them. But our main effort currently is on the use of glass, and we etch micrometer-sized channels or complex network of channels on the surface of glass. We isolate them, and then we inject different fluids, look at their interactions, etc. So here's an example, top left of the slide, of a sample from a formation in Mississippi in the United States, where there is an effort to create a commercial hub for CO2 sequestration. This sample then is looked at under a microscope. We examine it very carefully and decide on a portion of it that we think is representative of the overall sample. Then we rely on scanning electron microscopy to collect a mosaic of images covering a significant portion of it with nanoscale resolution. Then we develop algorithms to do feature detection that are common between adjacent images. And using this feature detection scheme, we are able to line them up very carefully and accurately and create a picture of the entirety of the part of the domain that we were looking at and investigating with nano resolution images. So with this, then we have a complex network that we can base our map on, and then the map that gets etched on the surface of a piece of glass to create a fluidic system. The issue is that the network inside a natural porous medium are in 3D. When we take a 2D image of it, the connections aren't there. So what we do is we collect data regarding the connections from a three-dimensional sample, and then we map that onto the two-dimensional image that we just created. Once we have figured out the connections and we've honored those connections, the connections that exist in the three-dimensional sample, onto the two-dimensional map, then we take that map and then etch that map onto the surface of the glass, create a device where we can use for experiments. We characterize the network by looking at their depth using confocal microscopy. And once we have a sense of their depth and we are happy with the way they look, then we create the device and do experiments in a complex system of equipment, depending on the exact experiment that we're doing. On this slide, you'll see on the left a schematic of the etching process that goes through a series of steps to take that network and transfer it to layer of photoresist, layer of chrome, and then the glass. And the etching that I just described is using a UV source that we have developed ourselves in the lab. It's homemade, and we have a good sense of the strength of the UV source that's shown on the top right. And then we take the devices at the very end before we make the microfluidic system. We take the glasses, we sandwich them together, we put it in a furnace, we raise the temperature close to the melting temperature of glass, and we bind them. And now we have a system we can look at. It goes under a camera that's high resolution. It's able to look at fine features. At the same time, it can look at the entirety of the medium. So we take an image of the entirety of the complex network, And we have the resolution to zoom in after the fact and see what happens inside the pore spaces. If we want to focus a little more on the details, we can place the device in the field of view under microscope that's coupled with a camera, a fast camera to collect images at a fast clip and look at the evolution of various fluids as they move through the porous medium. So imaging is a mixture between the two, a high resolution camera or a camera that's got the ability to collect images at a high clip, and it gets coupled to a microscope. For example, we can do particle image velocimetry when we're looking at the movement of microspheres that are little spheres that are buoyant inside the fluid, and their diameter is 1 micrometer, a millionth of a meter. And they move along, and we're capturing images at a fast rate. And then we can look at their movement and see what happens to the velocity, what happens to the fluid as it moves through this complex network. We also look at multiple fluids that are immiscible. They don't mix. Think of oil and water or a gas and a liquid. And what happens at the interfaces as they move through the complex medium. And you'll see on the right-hand side that exact question, trying to be exact with the location of the interfaces and trying to understand the evolution of the interfaces as it moves through the medium. With the high resolution camera, we can take an image of the whole medium and look at also at the same time. After the picture is acquired, we can look at small details inside the pore spaces to look at the evolution of a fluid moving another fluid out of the way. Now think CO2 sequestration storage. As you inject the CO2, it moves the existing fluid out of the way. We'd like to know how that happens and how to improve the displacement so that we have a more reliable system. We enhance the storage capacity and understand the safety parameters. And so it's important to map the flow regime and understand as a function of the parameter space, the properties of the fluids, et cetera, what happens. One of the areas of interest is understanding CO2 in the context of the subsurface. One of the things we look at is to create CO2 foams. Foam is similar to what persists at the bottom of a cappuccino cup. We have bubbles of air in that case that are capsules of of air. And what makes those bubbles persist for a while is basically related to the constituents in milk that's used to create cappuccino. In our case, we have CO2, and the interfaces are being strengthened by different surfactants or nanoparticles. Nanoparticles are small pieces of matter that are 10, 15, 20, 50, 100 nanometers in diameter. They then are, they position themselves at the interfaces and strengthen the bubble and allow them to persist for long periods of time, which creates a foam or a complex fluid that doesn't want to move as fast. So its ability to displace the resident fluid is improved. We enhance the storage capacity for CO2 in the subsurface. And then we use the microfluidic platform to look at that. So there's an example here on the screen. The top left is where we are injecting CO2 in the representation of that formation that I mentioned from Mississippi to see how CO2 is able to displace the resident fluid. On the right, instead of CO2, we're using complex fluids, essentially bubbles of CO2 that are enforced with nanoparticles along with soap, surfactant, to see how the improved system is able to displace the resident fluid, enhance the storage capacity. Again, this is very directly related to movement of fluids such as CO2 in the subsurface or H2 in the subsurface or petroleum systems. This understanding is crucial in engineering those systems very effectively. And we look at the use of different nanoparticles, for instance, silica, nanoclay, iron oxide, fly ash, et cetera, and each of them have their own interesting questions associated with them. We also look at the nanoparticles at a really small scale. So there is transmission electron microscopy that allows us to look at their aggregation. We're talking about nanometers here, 50, 20, 30 nanometers, to see how they come together and our ability to keep them apart and allow them to be effective in stabilizing these foam systems in the subsurface. The fluidic system allows us to look at various depths of channels and how that affects the movement of fluids. If you have channels sitting next to each other, one is a lot deeper than the other ones, what happens? And this is something that's directly relevant to the subsurface, where we have streaks of highly permeable regions sitting next to areas where everything is really tight. And we'd like to be able to engineer them so we utilize the entirety of the space and we don't have thief zones that just establish a connection and we leave behind all the pore volume that's sitting down in the subsurface for storage of CO2, for instance. We can use the fluidic systems to look at the interaction between the fluids. For instance, here's a video showing a fluid going in across a constricted equivalent to a pore throat, and how bubbles form and how the bubbles move. So it allows us to think about questions related to the interfaces and how the interface strength and resilience and viscoelasticity has the potential to affect the movement of fluids in these complex confined systems. We then can explore, again, questions related to the interface by designing complex channels and complex maps here on the screen. This is a system we designed to look at interfaces, again, in a two-phase system that don't mix, think oil and water, and how that then translates into the movement of fluids as a function of the interfaces strength and how that then informs the physics of fluids in the subsurface. Here's another picture where there are two fluids that coexist in this complex permeable medium in a microfluidic device, and we're interested in understanding the interfaces. Now, all this understanding the physics is allowing us to be predictive. Ultimately, that's the goal. We want to understand the physics, translate that understanding to mathematical models that then we find solutions to, and then we complete the circle by comparing the solutions to the experimental observations. And we are interested in doing this across scales. So for instance, the nanometer pore spaces where the flow is really complex, we'd like to be able to understand that. And on the screen, bottom right, you see a characterization at the nanoscale of a series of pore spaces inside a sample from the subsurface. And recognizing we have different flow regimes in these very, very confined spaces, we model and simulate movement / transport of a fluid inside this complex network, complex geometries, recognizing that the behavior of the matter changes in these confined systems. We then look at the impact of complexity. As you see on the right-hand side of the screen, we are looking at a larger, more complex system. On the left-hand side, we're looking at complex geometry, but very, very simple systems. So we combine this understanding, and eventually we'd like to look at large-scale systems, which is what you're seeing on the screen. This is a representation of a rock from the subsurface at a large scale, and we'd like to see the mixing between two fluids, one being pushed inside, and what happens between the injected fluid and the resident fluid and how they mix. And it seems like a flame of the inner, kind of how you might imagine a flame, but it's not. We have two fluids and the mixing is really complex, and that's what this represents. We'd like to be able to understand that. And ultimately, our goal is to be predictive with these models. And so we simulate this across a range of parameters and compare it with experimental observations. So here you see on the screen a depiction of a tomography-based system that collects information from the distribution of fluids in a sample of a permeable medium. And we then compare our predictions with the experimental observations, and hopefully they match in the sense that the predictions are accurate, and which in this case they are. You see the experimental observations in red and the predictions of the models in blue, and they line up pretty well. And so with that, thank you. I appreciate your attention. And here's the list of different entities that have provided support for this work. Thank you.